In our last lesson, we learned how there are lots of molecules that a cell needs. Molecules that a cell takes in, uses, builds, breaks down, and gets rid of. In this lesson, we're going to take a look at the cell membrane, because the cell membrane is the structure that controls the entry and the exit of those molecular materials. And we're going to see that the structure of the cell membrane makes it very well suited to do its job. Here's an overview of the cell membrane. So what we've done is we've taken the cell and we've sliced it open. And if you were to just look at this part, here's what the membrane would look like. You've basically got two layers, a bilayer, made up of phospholipids with some proteins and other things embedded inside. And this structure enables the cell membrane to do three things. First of all, it just forms a boundary between the cell and the environment. It also is selectively permeable, so it picks and chooses what materials enter and leave the cell. And finally, it's able to communicate with other cells due to some very specific components that we'll come back to later. Let's take a closer look now at that phospholipid bilayer. If we zoom in here on two phospholipids, this is what they would look like up close. And you can see that on the outside, we've got phosphate heads, which are hydrophilic. They like water and water likes them because they're both polar. Meanwhile, on the inside, we've got the hydrophobic fatty acid tails. And these guys are sandwiched inside since they don't like water. And this chemistry enables it to be a barrier to large molecules and polar molecules. So if you're large, you're not going to squeeze through the bilayer. Imagine this is a large molecule. Use your imagination. And if you're polar, you're not going to squeeze through the bilayer because those fatty acids will repel you. However, if you are small and or nonpolar, then you might be able to squeeze right on through because the fatty acids like you and you're small. Now, in terms of fatty acids, there's a couple different types that we haven't learned about yet. Some fatty acids, like these, have only single bonds in their hydrocarbon chains, and that makes them fairly rigid and solid at room temperature. And these are the types of fatty acids you get from eating animal fat products. Then you've got unsaturated fatty acids. Unsaturated fatty acids have some double bonds in their hydrocarbon chains. And this makes little kinks in the fatty acids, and it makes the fatty acids more fluid. So these will tend to be liquid at room temperature, and these you often get from vegetables. And in general, it's good for your phospholipid bilayer to have some kinks, to have some fluidity. Now, phospholipids aren't the only thing in the bilayer. There's other molecules embedded in there that are very, very important. If you look at this diagram, we've got lots and lots of different proteins. In fact, the cell membrane has over 50 different types of proteins. There's also little carbohydrates stuck on some of the proteins and stuck on some of the lipids. If they're stuck on lipids, then they're glycolipids. If they're attached to proteins, then we call that a glycoprotein. There's also molecules of cholesterol embedded in the cell membrane. So let's now take a look. Actually, before we take a look, one more note. Outside of the cell, there is a network of fibers known as the extracellular matrix. And this basically anchors cells to other cells. It's a framework. And then inside, there's another framework known as the cytoskeleton. Both of these are made up of proteins, and they provide support inside and outside the cell. So let's take a closer look at cholesterol. Contrary to popular opinion, cholesterol is not always bad. In fact, in the cell membrane, it keeps it at the proper fluidity. So if it's really cold, it helps keep the membrane fluid. And if it's really warm, it helps stabilize the membrane. So the membrane is kind of like Goldilocks. It doesn't want to be too fluid, but it doesn't want to be too stiff and solid. Now let's take a look at all the different proteins found in the bilayer. We're going to go through these one by one. Some are very familiar to you, some will be new. The first protein we're going to look at is the integrin. And as you can see from this diagram, the integrin spans the entire membrane. So you can see one end of it inside and one end of it outside. And it attaches to the extracellular matrix on the outside and the cytoskeleton on the inside. And that makes it very good at providing support for the cell membrane. You'll also note that the inside and the outside of the protein, the parts that face water, are made of hydrophilic amino acids. We call those hydrophilic domains because they need to interact positively with water. Meanwhile, the inside of the protein, the part that's embedded in the fatty acid bilayer, is made up of hydrophobic amino acids. We call that the hydrophobic domain. And we're going to see this in many different types of proteins. Enzymes are something we're familiar with. They catalyze chemical reactions by binding the substrate at the active site to produce a product, which could then become the substrate for a different active site and become a different product. And again, we see these enzymes span the membrane, so there's hydrophilic domains on the inside and outside, and hydrophobic domains in the middle. Transport proteins we've seen before, and they do exactly what they're called. They help those large molecules or polar molecules get through the membrane. So if you're too big or too charged to squeeze through the fatty acids, you find yourself a transport protein. And sometimes it might happen naturally without energy, and sometimes it does take energy, but more on that later. There's also a specific type of protein called an aquaporin, and these are proteins specially designed for the diffusion of water. Again, we can see the different domains depending on the chemistry of the cell. Receptor proteins, we are very familiar with these because they're part of the signal transduction pathway. So the receptor protein's job is to bind a ligand, some sort of messenger molecule. Could be a growth factor, could be a hormone. And then once it binds to the receptor, there's a conformational change, and that triggers some sort of signal inside the cell that may go off and interact with relay proteins and second messengers and transcription factors and that good stuff. Again, we can see the inside and outside facing water are hydrophilic amino acids with hydrophilic R groups, and the inside that's facing the fatty acids is full of hydrophobic R groups. Glycoproteins, this is a new one for us, and they're very, very critical. It's new, but actually we have seen them before, you just didn't know it. So glycoproteins have these carbohydrate chains attached to them, and those carbohydrate chains act as a little ID tag for the cell. They basically tell other cells that, hey, I'm one of you guys, I'm friendly. If it's a white blood cell, please don't eat me, I'm not a pathogen. And this is the same thing that glycolipids do as well. And we have seen these before when we studied blood types. The A and the B and the O blood types are due to the type of glycoproteins on a person's red blood cell. So if you have blood type A, it's because you're expressing the A-type glycoprotein in your membrane. And if you have blood type B, it's because you're expressing the B glycoprotein in your membrane. And O, you have neither, and AB, you've got both. So let's close by just stepping back and taking a look at the whole cell membrane. We can see there's lots of different chemical components interacting. Here's our cytoskeleton attaching to integrins, attaching to the extracellular matrix. We've got some glycoproteins, glycolipids, the phospholipids themselves, cholesterol, and then various other proteins doing various things embedded in the membrane. It's important to remember that this membrane is not static. It's not just sitting there, it's fluid. And if you check out one of the animations recommended on the website, you can see a better representation of how fluid the membrane is, because the proteins and the phospholipids can actually slide around. It's also important to remember that different cells, which have different functions, thus have different membrane compositions. So a red blood cell's cell membrane is not going to look exactly like a white blood cell's cell membrane or a neuron's cell membrane.